Regardless of which carbon fixation pathway a plant employs, rising atmospheric carbon dioxide concentrations will improve its water use efficiency. That is, rising atmospheric carbon dioxide concentrations will increase the amount of carbon dioxide assimilated into carbohydrate per water lost. This is a consequence of decreasing the disparity among the concentration gradients for water and carbon dioxide between leaves and the atmosphere. For the previous example of 25 degrees Celsius and 50% relative humidity, a doubling of carbon dioxide concentrations diminishes the concentration gradient, driving water vapor out of a leaf from 42 times to 21 times that of driving carbon dioxide into it. Plants might respond to these more favorable conditions in two ways. Strategy 1. They might decrease their stomatal apertures to conserve water while maintaining similar carbon dioxide concentrations within their leaves, and thereby assimilate carbon dioxide at similar rates. Strategy 2. They might open their stomata to the same extent and lose water at similar rates, but increase carbon dioxide concentrations within leaves and thereby accelerate carbon dioxide assimilation. So what do plants actually do? Before revealing the secret life of plants, I digress to some experimental methodology. In studies of plant responses to elevated carbon dioxide concentrations, plants are grown in controlled environment chambers, open-top chambers, or face plots, free air carbon dioxide enrichment. Controlled environmental chambers maintain light, temperature, humidity, and atmospheric carbon dioxide concentrations at designated levels. Such chambers consume large amounts of electricity to power lamps as well as to cool a brightly lit, confined area. Energy constraints thus limit light levels to nearly all chambers to a fraction of full sunlight. Open-top chambers have sides of transparent plastic film and large fans that blow various CO2 concentrations into the chamber as well as keep temperatures inside and outside the chambers more similar. Carbon dioxide is about 40% denser than air, and so any carbon dioxide added will remain near the ground until diminished through plant carbon dioxide assimilation. Unfortunately, environmental conditions near the chamber sides differ in light, temperature, and humidity from those near the middle of the chamber. A free air carbon dioxide plot, a face plot, consists of a ring of 24 to 32 vertical standing vent pipes set in a grassland, agricultural field, or stand of trees. Wind speed and direction and carbon dioxide concentrations in the middle of the ring determine how much carbon dioxide each stand pipe releases. This technique has proved effective over areas that range from a 2-meter diameter ring with a 1-meter tall standpipe in a California grassland to a 30-meter diameter ring with a 16-meter tall standpipe in a North Carolina pine forest. Unfortunately, such operations are very expensive. Supplying just the carbon dioxide for one large ring costs somewhere around $20,000 per month. For this reason, most of the elevated CO2 treatments in a face plot control carbon dioxide at about 0.05%, and sometimes only during the daytime. No matter what system is chosen for exposing plants to elevated carbon dioxide atmospheres, it becomes a major undertaking, one that strains facilities, funds, and personnel. Compromises must be made that invariably limit the number of experimental replicates. Because most studies on plant responses to elevated carbon dioxide have few replicates, a statistical approach called meta-analysis has proved valuable. Meta-analysis combines results from many independent studies to increase experimental sample size and thus enhance the strength of statistical tests. Commonly, a meta-analysis presents weighted means in which larger studies or those of less random variation have a stronger influence on the final value than smaller studies or those with more variation. Here I present the results of meta-analyses that surveyed hundreds of independent studies. So what do plants actually do when exposed to elevated carbon dioxide atmospheres? In general, plants behave in a manner intermediate to Strategy 1 and Strategy 2. They close their stomata by 22% and thereby conserve water, but they also increase carbon dioxide concentrations within leaves and thereby achieve 32% faster carbon dioxide assimilation. As a group, C4 plants close stomata and conserve water, Strategy 1, to a greater extent than C3 plants, reflecting that carbon fixation in C4 plants is less sensitive to carbon dioxide concentrations than in C3 plants. Trees tend to conserve less water and enhance carbon dioxide assimilation to a greater extent, strategy 2, than grasses, legumes, forbs, or shrubs. This proportionally large number of studies on trees biases the mean value for C3 plants. This highlights a major problem with meta-analysis, that of publication bias. Meta-analyses compile the results of studies published in readily accessible sources, but such studies may not evenly represent all types of species, nor all valid studies on a question. Authors are much more likely to conduct research on model species and to submit for publication studies with positive results rather than those with negative results. Readily accessible journals are more likely to accept for publication studies based on model species and that have received positive results. Publication bias has become such a problem in evaluating drug efficacy that several prominent medical journals will no longer publish results of pharmaceutical research unless the research project was registered in a public database when it was initiated. In this way, negative results on drugs will not just disappear. Unfortunately, no equivalent system exists to track studies about the influence of global climate change on organisms.